Hello everyone. Let's get started. Today we're going to be talking about cellular organization and specifically the organelles. Remember that all cells are surrounded by a plasma membrane, and inside those cells there's a gel-like material called cytoplasm. Within that cytoplasm, all cells contain DNA, and they use that DNA as their genetic material. They also contain ribosomes, which are the cellular machines that are used to perform the process of translation. Today we're going to be focused on eukaryotic cells because eukaryotic cells are the cells that contain organelles and a cytoskeleton. We'll be talking about the cytoskeleton in detail later in the semester. Today, we'll focus on the organelles. When we translate the word organelle, it means little organs. So you can think of organelles being somewhat similar to the organs in your own body. Remember, your organs are built in a way that allows them to do a very specific set of functions for your body. Organelles are very similar. They are structured in order to complete a certain set of tasks for the cell. Organelles, by definition, are surrounded by a membrane, so they are membrane-bound compartments within the cell. Remember, there are two organelles that we believe arose through a process called endosymbiosis. Both the mitochondria and the chloroplast are believed to have evolved when a larger primitive cell engulfed a smaller primitive cell, and that smaller cell became part of the larger cell. The internal membrane of the mitochondria and the chloroplast was therefore the original plasma membrane of that original primitive cell, and the outer membrane of the mitochondria and the chloroplast was the plasma membrane of that larger primitive cell. We have evidence that the mitochondria and the chloroplast arose through endosymbiosis, including some structural features that differ between the inner and outer membranes of those organelles. Another piece of evidence for endosymbiosis is the fact that both the mitochondria and the chloroplast contain their own small pieces of DNA. There is another organelle that has two membranes, but they are not two separate membranes like they are in the mitochondria and the chloroplast. And that organelle is, of course, the nucleus. The nucleus does have a double membrane, but it's connected at certain locations, and so it's referred to as the nuclear envelope. The inner and the outer membrane come together and connect with each other at openings in the nucleus that are called nuclear pores. So we do refer to an outer membrane and an inner membrane, but just like an envelope has its two layers connected at the edges, the nuclear envelope has its two membranes connected wherever there is a nuclear pore. There is a space, albeit a small space, in between the outer and inner membrane of the nuclear envelope, and that space gets a name. It's referred to as the perimembrane space. There's also a layer of material that lies just within the inner membrane of the nuclear envelope. It's called the nuclear lamina. That term lamina is a generic sort of term that we use to basically indicate a layer that deserves our attention. The nuclear lamina is a layer composed of several different proteins that is right up against the inner nuclear membrane. It's very closely associated with that membrane, and it helps maintain the general shape of the nucleus. Within the nucleus, of course, lies the chromatin. And chromatin, if you'll recall, is the DNA molecule that has been wrapped around and carefully ordered with proteins called histones. Chromatin lies in a gel-like fluid called nucleoplasm. And sometimes under the light microscope, we can see darkened areas inside the chromatin, and we refer to those areas as nucleoli. The singular of that word nucleoli is nucleolus. You won't always see a nucleolus or nucleoli inside the nucleus of a cell, but when you do, you know that you are looking at a site where ribosomal RNA is being actively transcribed. On this slide, you can see a simple drawing of a nucleus. The nuclear envelope is shown in a deep purple color, and the nuclear pores are shown as dark blue circles. They've cut away a region of the nuclear envelope so we can peek inside. There's material that is shaded a lighter purple color that represents the chromatin, and there's an area that is dark blue that represents the nucleolus. And the artist has also drawn for us a couple of representative chromosomes. Now, it's important to know that chromatin is the dominant form that we find DNA in at most times inside the nucleus of a cell. Chromatin is dispersed throughout the nucleus, and when you look at it under a microscope, it looks quite homogeneous. There's one time that chromatin will condense and become visible in a completely different way, and that's when the cell is preparing to divide. It's at that point that chromatin becomes what we call chromosomes, individual bodies of DNA and histone proteins that we refer to as chromosomes. So unless the cell is preparing to divide, the DNA in the nucleus will be in the form of chromatin. On this slide, we can see a rather complex drawing of a piece of membrane. Now, this is not cell membrane, this is nuclear envelope. So if we look at the yellow colored areas, these represent the two layers of the nuclear envelope, the inner membrane and the outer membrane. Now, you can't really see it very well in this drawing, but the two layers of membrane are attached to each other right here on the edges of this large, very complex looking structure, which represents a nuclear pore. Now, it's important for us to know that nuclear pores are very complex. They are not simply holes in the nuclear envelope. The nucleus has an important job to do. It has to keep the genetic material for the cell isolated from the rest of the cell. It's protective, and therefore it's important that it keeps a tight control over what molecules can come into the nucleus and what can leave the nucleus. The nuclear pore contains these structures that are referred to as the nuclear basket and nuclear fibrils. These form essentially a gate in combination with the ridges that you can see here in the wall of the pore. Now, it's not important for you to know all of the terms involved in the pore, like annular subunit and cytosolic fibril. Instead, you should understand that the pore is gated. And what that means is it's essentially closed off to most molecules. Unless the molecule can interact with the pore and trigger it to open, it will not be allowed in or out of the nucleus. So the nuclear pore helps regulate the flow of material in and out of the nucleus. Now, the other thing you should note is that from the surface, the outer membrane of the nuclear envelope, the pore is a little bit raised up off the surface of the envelope, and it has these strange little hairs coming off of it. This is how it will bind with and interact with molecules that are trying to come into the nucleus through these external structures. The basket part of the nuclear pore is actually on the inside of the nucleus. The other thing to note here is this green mesh-like material that sits underneath the inner nuclear membrane. This is the nuclear lamina. And recall that this protein-based material is going to help the nucleus keep its shape. There are some other proteins that are embedded in the nuclear envelope, just like we see proteins embedded in the cytoplasmic membrane. One of the things that we need to be able to do when we talk about organelles inside of eukaryotic cells is learn how to recognize those structures as they actually appear in the cells. Now, when we look at most eukaryotic cells under the microscope, the light microscope, we can't see all of the individual organelles. With the most commonly used cellular stains, we can typically see the nucleus, but we won't see it in any detail. Sometimes you can see other organelles, but it's quite inconsistent and completely dependent upon the stains that you use. We actually require an electron microscope in order to see all of the organelles within a cell and to see them in any detail. Now remember, when it comes to electron microscopy, there are two commonly used varieties of that technology, what's called the transmission electron microscope and the scanning electron microscope. A transmission electron microscope is going to produce an image like the one on the upper left-hand side of the slide. A scanning electron microscope will produce an image like the one on the lower right-hand side of the slide. The transmission EM image will show the internal details of the cell, and the scanning EM image will show the surface details. So two very different types of electron microscopy are available to help us see two very different views of cellular structures. So let's start on the upper left-hand side and look at this TEM, or transmission electron microscope image of a cell called a lymphocyte. Now, lymphocytes are one of the types of white blood cells that are part of the immune system. This is the boundary of this lymphocyte, what I'm tracing right now with the cursor. You can see that the plasma membrane is not particularly smooth. It has some extensions coming out of it. The lymphocyte is actually quite a dynamic cell. Its structure changes somewhat from moment to moment. What I want you to note, however, is this really dark area within this lymphocyte. This dark area takes up almost the entire volume of the cell. And then there are some lighter areas shown within that darker area. This dark structure is the nucleus. And the dark and light materials inside the nucleus are different areas within the chromatin. The lymphocyte happens to be a cell that has an enormous nucleus relative to the size of the cell. And so it's a good cell for looking at the nucleus in detail with a transmission electron microscope. Now look at the SEM image, the scanning electron microscope image. This is the one again that shows us the surface. This one happens to be colorized as well. So they have colored the surface of the nucleus here on the bottom half of the image, a dark brown color. And structures that are lying outside of the nucleus are shown in a tan color. The blue arrows that are added to the image are pointing out nuclear pores. And as you can see, they are not symmetrically dispersed over the surface of the nucleus. They're not evenly dispersed. But you can also see that there are lots of them. And in fact, if you were able to visualize the entirety of this nucleus right now, there would be hundreds of pores on the surface. The next organelle is the mitochondria. Now, mitochondria are relatively large organelles. Remember when we talked about the relative size of different types of cells, we said that a bacterial cell is about 1/10 as big as an animal cell. And mitochondria can be as large as a small bacterial cell, typically in the range of 1 to 3 micrometers. So this is a prominent organelle inside a eukaryotic cell and relatively easy to locate, again, if you're using the right kind of microscope. The mitochondria is the site of a critically important metabolic reaction that we refer to as aerobic cellular respiration. Remember, this is the metabolic process that produces ATP. When we look at the structure of the mitochondria, it has two membranes around it, an inner mitochondrial membrane and an outer mitochondrial membrane. The inner membrane has a much larger surface area than the outer membrane does, and so the inner membrane is somewhat folded up. And we refer to those folds as crista, or in the plural, cristae. There is a space on the inside of the inner mitochondrial membrane, and that space is full of a gel-like material that's called matrix. There's also a space, although a smaller one, between the outer and the inner mitochondrial membranes, and that space is referred to as simply the intermembrane space. It's important for us to know the difference between these two spaces, because when it comes to aerobic respiration, different processes are going to be happening in both the matrix and the intermembrane space. Remember, mitochondria are believed to have evolved through endosymbiosis. And so there are two other important structures that will be found in mitochondria, and those include mitochondrial DNA and ribosomes. Both of those are located in the matrix. Mitochondria, therefore, have their own small piece of DNA, and when it gets transcribed and translated using those ribosomes, we see the production of proteins that will be used right there inside the mitochondria. One of the most interesting things about the mitochondrial DNA is that it has maternal inheritance. And what that means is that it is passed down from the mother directly to the offspring. There is no intermixing of maternal and paternal mitochondrial DNA. Instead, the DNA from the mother is passed down to all of the offspring. So the mitochondria is the site of this critically important metabolic process called aerobic respiration, or cellular respiration and oxidative phosphorylation. This is the place inside of a eukaryotic cell where carbohydrates like glucose will be broken apart or oxidized all the way to carbon dioxide. That process occurs in the matrix of the mitochondria. Remember that as the glucose is broken apart, as it's oxidized, individual covalent bonds in the molecule are being broken, and the electrons that are in those bonds will be released. Electron carrier molecules will temporarily receive those electrons and hold on to them until they can be transferred to the electron transport chain. The electron transport chain is located in the inner membrane of the mitochondria. It is embedded in that membrane. And if you recall, the ETC is simply a group of specialized proteins that will be used to harvest the energy from the electrons that are passed down it. Now, once those electrons reach the bottom of the electron transport chain, they are received by a final electron acceptor molecule. And in aerobic organisms, like human beings, that final electron acceptor is oxygen. As electrons are passed down the electron transport chain, the energy is harvested from those electrons, and it is used to build a gradient, a hydrogen ion gradient. Hydrogen ions are pumped from the matrix of the mitochondria across the inner mitochondrial membrane and into that intermembrane space. Once that gradient is in place, it's allowed to resolve or go away when the hydrogen ions flow back into the matrix through the molecule called ATP synthase. ATP synthase, like the electron transport chain, is embedded in the inner mitochondrial membrane. Cells are going to vary in how many mitochondria they contain. Any cell that has a very high requirement for ATP is going to have a greater number of individual mitochondria within it. For example, skeletal muscle is known for having many individual mitochondria inside of it. On this slide, we can see a diagram of a mitochondrion at the bottom of the picture, and we can see again an electron microscope image up at the top. This happens to be a TEM image, a transmission electron microscope image. Let's start with the drawing at the bottom. The artist is showing us an outer membrane in darker orange color, and then an inner membrane surrounding this lighter orange compartment. You can see that the inner membrane is folded up. It has a much larger surface area than the outer membrane does. The folds, again, are referred to as cristae. Inside the inner membrane, in this compartment that is most internal, this is the matrix. And within the matrix, you'll find ribosomes, indicated by these little dots, and mitochondrial DNA. In the space between the inner membrane and the outer membrane of the mitochondria, there is also a space. This is called the intermembrane space. Now, like a lot of cellular structures, when you look at a mitochondrion under an electron microscope, it doesn't look nearly as pretty and perfect as the picture does. If you look at this mitochondrion, for example, it has the same sort of shape as the mitochondrion in the drawing, but it has a little bit of a blip here that you wouldn't expect to see. And the cristae don't look particularly organized. It depends upon the cut, the section that is taken through the cell, as to what shape and size the mitochondria is going to be. You have to remember that when cells are prepared for viewing under the microscope, they're going to be sectioned, just like you would slice a loaf of bread. Mitochondria are three-dimensional structures, as are all organelles. And so depending on where the slice lands, you're going to see a different view, a different section through that organelle. So in this EM image, we can see cristae here. We can see some of these folds. We can also see the outer membrane and the inner membrane. Those have been labeled. In addition, we can see a different color to the mitochondrial matrix. It's much lighter on this preparation. On this slide, we have two more TEM images of mitochondria to look at. Let's start with the image on the upper left. This is the outline of the mitochondria. This is about as perfect a section as you'll ever get through a mitochondrion. You can see the outer membrane surrounding the organelle, and then the inner membrane making up these folds, these cristae. There are lots and lots of cristae visible here. The other thing that you can see in this image that we haven't spoken about yet are the folds of membrane that are located outside of this mitochondrion. This is rough endoplasmic reticulum, and we'll talk about it in just a minute. I also want to draw your attention to the transmission electron microscope image down at the bottom of the slide. This is an image that was taken through skeletal muscle. And I think you can see that there are these striped regions inside the muscle tissue. These are sarcomeres, the individual units of contraction of skeletal muscle. And they're present visibly throughout this image. But the reason I put the image in is because of this. These lighter areas that are visible in the image, these are individual mitochondria inside this particular cell. Now the outlines of this cell are here, then they go outside of our view, and then they continue over here, and then go outside of our view. So this, from here to here, this is a single muscle cell, or what we call a muscle fiber. And most of the volume of this muscle cell is full of these filaments that make up the sarcomeres. But there's still room for all of these mitochondria located in between the individual filaments of the sarcomeres. The nucleus of this particular muscle fiber is not in our view, unfortunately.